Hi everyone. Oh, this is quite embarrassing. Someone just alerted me that I had failed to upload this lecture and I had asked about it in the test. So that's really unacceptable and I really apologize. So what I'm going to do is obviously upload this lecture so you can have a listen, read through the notes. And for all the questions that I asked in test 2 that was about this topic, I will just remove it from the calculation. So I think everybody did really well, so I would be surprised if you didn't get almost full marks, most of you. So my mistake is your gain, hopefully. I will ask about this topic, I think, in greater detail for the third test, where we combine with Dr. Yahya's first part. So let's get into it. So this topic is the final topic for immunology, which is organ transplantation. So it's another immunological application. And if you recall, the previous application we covered was about diagnostics. And it's quite funny. I usually don't split the diagnostics lecture into two. I usually give it as a very long lecture, but I thought, because of the platform we have now, I can split it into two. And I think when I was doing the calculation of how many lectures I've covered, I mistakenly thought that was actually the second lecture for the diagnostics was actually the transplantation lecture. So very silly mistake again. But this is technically what should have been the second last lecture, basically. For this lecture, the learning outcomes include that students... Right, so where were we? Yes, so the learning outcomes are that you could describe the basic concept of transplantation and different types. Then explain how the immune system plays a role in whether or not a transplant is successful. And whether or not a transplant is successful is usually based on whether or not the transplant is accepted or rejected. And so the rejection involves the immune response. And finally, you should be able to explain the ways the immune system can be manipulated to improve this transplantation procedure. So before we go into all of that, let's have a look at what transplantation actually means. So transplantation is the process of taking a graft, which is basically an organ or a living tissue, and putting it in another part of the body or in another body completely. So you can do, you can move skin from one part of your body to another part of your body. For example, that can happen for burn victims, serious burn victims, or if you can't give yourself that part, then you can get a donor to give it. So this started in history in 1908, where Alexis Carrel interchanged the kidneys in a series of cats. So both kidneys he actually interchanged, and he noticed that some of them were actually still able to perform the function, which is peeing, having urinary output, and it was for up to 25 days. But then eventually all of the cats died. But that series of experiments essentially told or tells us that it doesn't matter if the organ comes from another body, if it's put in, it can resume its function. In 1935, the first human kidney transplant failed because of a mismatch between the blood types between the donor and recipient. And that actually resulted into the immediate rejection. And so this immediate rejection, as we'll learn later, is often mediated by preformed antibodies. Because if you remember the blood type mismatch, it's a very serious thing. You already have antibodies against it, so it would happen immediately. It wouldn't even take that long for a reaction to occur. So transplantation is actually a huge part of, I'd say, modern medicine. And there's a projection that it's going to grow by 10% by 2023. And this is primarily because as time passes, we have more and more chronic diseases. We have more research that actually improves some of these outcomes. And then we also have a lot more money to spend on these otherwise very, very expensive procedures. So this makes transplantation more of an option for a lot of people who need to replace some organs that may have been damaged due to certain chronic diseases. But that said, there's still a lot of challenges in transplantation. So this is a headline from about 3 years ago. I wasn't able to find anything more recent, but that pretty much says that about 21,000 Malaysians were on the waiting list for organ donors. This list here is from 2019, which I showed the class last year. And this is the American data, or data from the US. I wasn't able to find one for Malaysia, but you can see the number is quite large, and the number one organ that a lot of people are waiting for is kidney. And in 2020, the rank has not really changed, just the numbers have changed a little bit. Some have gone down, some have gone up a little bit. But by and large, the kidney is the most high in demand organ that people need to get replaced. So there's the challenge of actually having the organs for people who need them. And then there's also the rejection of the transplant. So even if you end up going up on that waiting list and you're able to do the transplantation, doesn't mean it's going to be successful. So there's a couple of challenges there. And about the rejection, we know it's mediated by the immune system, so some immunosuppressive agents can actually be used to prevent or delay the effects of rejection. So what kind of types of transplants are they? So we've got obviously we've got different types of organs, but we usually categorize transplants whether or not they came from the same individual. So that's called an autograft. So like I said just now, for example, if you have a very significant burn, you can actually transfer some of the skin from another part of your body to the part that's burned. Or you could have an isograft. So that's when you have tissue transferred between genetically identical individuals. So it's like identical twins. Or if you have animals that are inbred and are the same strain, they're also considered isografts. So they're actually genetically very similar. Sorry. And for allografts is when the tissue is transferred between genetically different members of the same species. So from human to human, but obviously not a genetically similar human, just species similarity. And the last one is xenograft. So xenograft is when you get an organ or you transfer an organ from a different species. So for example, the heart of a baboon to the heart of a human. So this picture, I think, encapsulates it quite clearly when you have an autograft, an allograft, an isograft, or a xenograft.
Mom,
so the main issues that arise are... Right, so the immune response typically is a problem when you're doing an allograft. So that's when you try to take an organ from the same species, but somebody who's genetically different. So autografts and isografts are usually accepted because the body pretty much thinks it's the same person. So it's not going to reject it. Xenograft would obviously be worse than the allograft. But it's not as common. Allografts are much more common. So the rate of the allograft rejection depends on the type of tissues that are involved. So typically skin grafts are rejected faster than other tissues such as kidney or the heart. And the whole idea of the immune response in graft rejection is based on the idea that we talked about earlier, which is the specificity and memory. So it goes on two steps. So the first step, or the first set rejection, occurs, for example, if an inbred mouse of strain A is grafted with skin from strain B, and then they'll get a primary graft rejection. So let's say it's not compatible. But the second set rejection is because of the memory that was obtained. So when the second strain B graft was transferred to a previously engrafted strain A mouse, then the antigraft rejection will occur even faster. So a complete rejection will occur within 5 to 6 days instead of the length that it takes for the first set rejection. But I think this picture is a better way to demonstrate that. So let's say in these columns you've got 3 different scenarios. So the first scenario is if you actually put in the graft and it's accepted. So let's say it's an autograft. So it's a skin tissue from the same person, let's say to the burn area. So you've put in the skin there, and then the blood vessels start to vascularize, revascularize, and then by a week it starts to heal. And by a couple of weeks or 2 weeks, it's essentially successfully resolved, and then it's accepted. Now in this case, in B, you have a grafted epidermis, perhaps not from the same person, and you observe a first set rejection. So what happens is around day 3 to 7, the first week, there is still some revascularization, but instead of it resolving, after day 7, you actually start having cellular infiltration. So that's the immune cells coming in, attacking that graft, and essentially killing it. So by day 14, instead of resolution, you actually have thrombosis and necrosis and blood clots and damaged blood vessels, and basically it didn't work. So it was rejected. It's not accepted to join the rest of the skin. I'm doing a lecture, okay, Amber? Can you stay over there? So the third column is when there is a second set rejection. So the second set rejection, remember, there's already some memory against the graft. So in this case, let's say you try to graft again. So this will be rejected even faster. So by 3 to 4 days, you basically start seeing that infiltration with all the mediators, and so the necrosis and thrombosis occur by day 5 or day 6, and that's basically it. So by day 6, you're already seeing that rejection occur, because if you remember, even when we talk about vaccination and memory, you'll have a much lower response that's a little bit later, but in the secondary response, it'll be much faster and much higher. So this is basically the same effect that you see, but this time not against a foreign invader. Well, it is sort of a foreign invader, but it's not really an invader. It's a graft that the immune system rejects. So a key thing that we know now is that the T cells play a very big role in graft rejection, and there are a few studies or experiments that clearly highlight this. So basically one of them is that when mice that don't have thymus or any functional T cells, they're actually unable to reject any allografts or even xenografts, so they're able to accept it readily. Another point is that the T cells from an allograft primed mouse are actually shown to transfer the second set allograft rejection. So that means if, let's say, you have in this case, this mouse A has already experienced the first set graft rejection. So they transplanted the T cells from that mouse to mouse B, and then the mouse B actually demonstrates a second set rejection, even though they haven't had the first transplantation, which means all of that memory is coming from the mouse that had that first rejection. So the other point is that just gonna move my face. Okay. Yeah. So is that if you actually inject monoclonal antibodies to deplete the CD4 cells, you are actually able to improve the survival of the graft, because obviously the CD4 T cells are not able to recognize the receptors and able to activate. So these are some very strong suggestions that the T cells play a very, very big role in graft rejection. So the whole idea of the rejection and how they recognize whether or not something's coming from a different host is this histocompatibility. So a lot of the antigenic loci responsible for the allograft rejection is located in the MHC. So it's called the H2 complex in mice, and in humans it's called the HLA, so the human leukocyte antigen complex in humans. And these are inherited, and the children can children can accept the graft from the parents, but not vice versa. So this means, although a child should be able to accept a graft without rejecting it from the parents, it's harder for a child to donate to the parents without some risk of graft rejection. And that's because the child does not have exactly the same genes as the parents, but the parents don't have the exact same genes as the child, but the child would have gotten their genes from the parents. So it's basically about a 50% match, because you'll have the other parents' genetic makeup within the child that would then not be recognized by the other parent. Yeah. So this basically is just about a 50% match instead of a 100% match. And between siblings, you would assume even a lower match, because there's also different combinations that could occur. So the choice of donors is really important, and the first one is obviously the blood type compatibility, but also the major histocompatibility complex, and that's where the most intense graft rejections can occur. So that's why the first thing you do before you do any transplantation is to check if they're actually from a suitable blood group. Otherwise, it'll obviously be a very hyperacute rejection. So the other one is they'll screen for the MHC and the HLA compatibility, and often you kind of try to improve your odds of being successful. So start with the closest relatives, like the parents or the first order siblings, and then they'll do tissue typing, which tries to sequence and establish that the HLA alleles are similarly expressed by the recipient and the donor. Or they'll try to do cross-matching and to see if there's any preformed antibodies against the potential donor, just in case that will show that the graft will be rejected. So 80% of kidney transplant patients with a positive cross-match often have immediate transplant rejection. So and there's only about 5% of patients with a negative cross-match that has this outcome. So it shows that it's quite a useful way to screen whether or not a graft might be rejected. Then there's also the Luminex cross-matching assay, where they use microbeads that have fluorochromes of different intensities to carry a different HLA protein. And when the serum from the person who's supposed to accept the graft is incubated with these beads, any antibodies binding will be detected with another antibody that's labeled, the secondary antihuman immunoglobulin, and then this laser excitation will then be detected and used to see whether or not the associated HLA molecules react with the serum. So that also gives you an idea. However, even when the MHC antigens are identical, there can still be a possibility of rejection, because there are also other loci or genetic differences that play a role in the graft rejection, so including the minor histocompatibility locus. So the MHC antigens are usually recognized directly by the T cells based on their alloreactivity, but the minor histocompatibility antigens are only when the peptide fragments are presented in the self-MHC molecules. So even though the rejection is less intense, it still can happen, and it can still lead to a graft rejection. So even though if you have a very strong HLA match, they will usually still give the patient or the recipient some immune suppression just to reduce the risk of graft rejection due to these other loci, such as the minor histocompatibility antigen complexes. Right, so let's go into graft rejection. So we kind of saw the difference between the acceptance, first set rejection, the second set rejection. And so the idea is that the graft rejection occurs in stages, and it can, we also talked about how it's mainly mediated by T cells, but it involves both, really, because if you recall that preformed antibodies will also play a role. So it depends on the alloantigens expressed on the cells. So in the first stage, a sensitization occurs. So that's right after the transplantation, and then the antigen-reactive lymphocytes will start to activate and proliferate. And then the effector stage is just when the immune system tries to destroy the graft. So the sensitization phase is when the CD4 and CD8 cells recognize the alloantigens expressed on the cells of the foreign graft, and then so they get stimulated, activated, and then they will expand. And they'll recognize both either major or minor histocompatibility alloantigens, and they'll either directly recognize the donor MHC molecule, or they'll recognize the peptides in the cleft of their own MHC molecules. And usually the dendritic cells are going to be found in most tissues that express a high level of the MHC2 molecules. So the activated allogeneic donor
I don't know.
So the activated DCs will mediate the presentation in the grafts or in the lymph nodes that they drain to. And the APCs of the host origin can also migrate into the graft and endocytose the foreign alloantigens. And the transfusion of the donor blood into the graft before the transplantation can actually help, I guess, in a sense, get the donor, the acceptor or the recipient ready to accept the graft. And also, if you recall, because when you give these signals, they give the antigens without the co-stimulation, which was asked in the test, it actually induces tolerance or T cell energy instead. So that's one way that they start trying to reduce the intensity of the immune response to the graft. So that was the the sensitization stage, and now it's the effector stage. So again, this is when you start really seeing the cell-mediated responses. So you'll have an influx of the immune cells like we saw in the diagram. We'll see T cells, we'll see APCs, and then you'll pretty much see the late-type hypersensitivity response. And the cytokines that are made will just start will mediate the graft rejection and cause inflammation and promote more T cell proliferation, more DTH responses, and then the B cells will also start making antibodies against it, particularly the IgG, which, as we recall, will also be able to activate the complement system. So there are some cytokines that we know are more closely associated to graft rejection. So actually targeting these cytokines can help reduce the graft rejection. For example, neutralizing interleukin-17 has been shown to extend the survival of cardiac allografts in mice. So those are the more common reactions that you see in the effector stage, but there are also less common mechanisms. So this includes antibody-mediated complement lysis and destruction through ADCC. So this involves the T cell maintenance of alloreactive B cells. So the alloreactive B cells continue to activate the complement through the antibodies, which then deposit in the endothelial cells lining the graft capillaries. So that can also occur, and it can also occur in an earlier stage. So this basically is a pictorial summary of all the effector mechanisms that I just mentioned just now involved in the allograft. So you've got the APCs, you've got the T cells, you've got the DTH T cells, activated macrophages. You also have the B cells, complement activation, lysis through complement, antibody-dependent cell. Forgot what the other C is. Really sorry, guys. Sunday evening. So membrane damage, lactic enzymes from the macrophages, and that delayed type hypersensitivity that you would see. So a few ways that the graft really gets destroyed by the immune system. So again, the idea of the graft rejection happens in stages, and it can either, so when a transplant occurs, the surgeon pretty much has to monitor at different time points, just because it's still okay. In the first 24 hours, you're still not clear yet. So they have to monitor a bit longer because it can happen at different stages. So the first one is the hyperacute rejection, and that will happen within the first 24 hours after the transplant. But usually this can be avoided because, there'll be all that screening test to make sure that the HLA matches, et cetera, et cetera. The second stage is the acute rejection. So that happens between 7 to 10 days post-transplant. And then finally, the chronic rejection can also happen. So even though, let's say, you've passed the first 2 weeks and it seems like the graft has been accepted, there are cases where months or years after the transplantation, you start seeing the immune system rejecting the graft as well. So the first one is obviously the scariest one, the most intense one. It will happen very fast. And this is because it's mediated by pre-existing host serum antibodies for specific antigens from that graft. So it could be that there's already been repeated blood transfusions that have induced antibodies to the MHC antigens that are expressed on the allogenic white blood cells in the blood. Or, for example, if there's been repeated pregnancies. So the woman has actually developed antibodies against the father's set of antigens in the fetus, or if there's been exposure to infectious agents. So the MHC cross-reactive antibodies are the ones that are going to be mediating the hyperacute rejection, or if there's been a previous transplant, and so that's already induced the antibodies to the MHC antigens on the graft. So once the HLA antigens bind to the endothelial cells of the graft, immediately the complement system will be activated, and there will be an accumulation of neutrophils, and you'll see the inflammation and endothelial damage, the obstructive blood clots. So it'll get destroyed very fast. So I think this picture will be more clear than what I just said. So you've basically, the key thing is here, there are already antibodies that are existing in the host, in the recipient. So let's say, for example, the recipient receives a kidney, and there are antibodies against whatever is on that kidney. So it will start entering and start mediating the complement activation, then all the complement will start attracting more neutrophils, but then start producing lytic enzymes, and then you'll have a destruction of the endothelial cells, platelets, and then you'll get clots, which will obviously damage that organ. So as I said, usually we have tried to avoid the hyperacute rejection phase because there's already been screening done. So the next one would be the acute rejection phase. So this is mediated by the T cell responses. And this is categorized by the infiltration of macrophages and lymphocytes to the site of the tissue. So this occurs, this is better explained by that effector stage that we just saw in a few slides ago. So this one is the one that's, I guess, a bit harder to manage because it can take quite some time. So this involves both the humoral and the cell-mediated responses by the recipient. While you can use immunosuppressive drugs to help you prevent the acute rejection, for example, it's harder to do this for chronic rejection because you don't want somebody to keep on taking immunosuppressive drugs. They have their own adverse effects, so this won't be a way that you can manage it. And this actually can happen in up to 60% of cases, where there's some form of chronic allograft dysfunction, and you'll see antibodies against the donor's antigens in the recipient. So this is suggesting that the chronic phase is mediated more by antibody-mediated rejection on top of cell-mediated responses. As I mentioned before, the key way to manage a transplant or to reduce the risk of a graft rejection is by using immunosuppressive therapy, because we know this is all basically a product of the immune system trying to do its job, but in this case we don't want it to do its job because we want to bypass nature and use modern techniques to improve our lives. So you can use drugs that can suppress the immune system, and it can either be general or specific. And there are problems with the general one, typically because you know that once you suppress the immune system, obviously you're suppressing your defenses against other things, so you're more likely to get sick from infectious disease. But it also increases the risk of cancer, because we know the immune system plays a big role in controlling cancer as well. And these immunosuppressive therapies can actually be quite aggressive as well. So there's some where they completely irradiate the lymphoid to completely eliminate the lymphocytes. Or it's more drug-based, like using immunosuppressive drugs. And then there's also the more specific immunosuppressive drugs. So the X-ray irradiation is used just before you get the transplant in a total lymphoid irradiation. And this is usually for bone marrow transplantation or to treat graft versus host disease. So graft versus host disease is very interesting. You can read up on it on your own, but this is where it's kind of like the reverse. So before this, we were just talking about the host or the recipient trying to kill a graft, right? Because, the host is the bigger being. The graft is just a small addition to the body. So the host will then try to eliminate the graft if it doesn't like it or if it doesn't match. In graft versus host disease, as the name suggests, that little graft is the one that's trying to kill the host. And basically it's bringing in its own lymphocytes and starting to attack the recipient's tissues. If you ever watch Arrested Development, there's a really funny episode about graft versus host disease in one of the characters who got some hair transplants. So he's bald, and he got hair transplants, and the hair was trying to kill him, basically. But he had beautiful hair. So it's a comedy, a bit of a satire. So with this type of condition, you use total lymphoid irradiation to treat, or you use it to prevent any problems with bone marrow transplantation. So there will be multiple X-ray exposures to the thymus, the spleen, and the lymph nodes before the transplant. So the recipient is extremely immunosuppressed under this state. But the bone marrow is not irradiated. So this means once the transplant is over, or they're fairly confident that the graft has survived, the bone marrow can replenish the lymphoid stem cells and renew the population. And they tend to be actually more tolerant to the antigens of the graft. So this is basically the sites where the irradiation will take place. All right, so the other one is using drugs, right? So just now it was radiation, so this one's using drugs, and it's generalized immunosuppressive therapy. So usually they use mitotic inhibitors along with the immunosuppressive drugs, and immunosuppressive drugs that are general, if we recall, tend to be corticosteroids. So one example is azathioprine, which is given before and after the transplantation to suppress the BMT cell proliferation, or cyclophosphamide, which is an alkylating agent that disrupts the DNA chain, so it reduces the cells being able to divide, and it also blocks T cell proliferation. And either of these will give you some pretty serious side effects that could affect your gut and your liver. But it does increase the survival rate of the transplant. So then you also have specific immunosuppressive therapy. So this is a bit better than the general one because it is more targeted and it tries to inhibit the immune response to the alloantigens on the graft. But it tries to still allow the host or the recipient to respond to other foreign antigens that could actually be dangerous to them. So these targeted immunosuppressive agents usually involve monoclonal antibodies as per normal, and they usually target the cell surface molecules. So one limitation is that the monoclonal antibodies, as we know, usually comes from animals, usually the mice. So the recipients will develop an immune response to the non-human epitopes, and they'll clear the monoclonal antibodies from the body. However, now there's a way to humanize the monoclonals, and you'll have mouse-human chimeric antibodies that are more tolerated by the host. So they'll work by depleting the recipient of a particular cell population. So it doesn't suppress the entire immune system, but just certain cell populations. Or it'll block a key step in the immune signaling. So there are some examples there, but the example I asked in the test is the rituximab, so the monoclonal antibody against the CD20. So that helps suppress mature B cells. There are also some monoclonal antibodies that block co-stimulatory signals, and so the graft is prolonged. The survival of the graft is prolonged by blocking the CD80 signal. And so this is a new drug called belatacept, shown to induce energy in T cells. So if you block the co-stimulation, then the T cell just becomes energetic and tolerant instead of activated. So this is a bit more of a complex figure, but it actually summarizes quite nicely what are some of the targets under specific immunosuppressive therapy targeting the T cell signaling. And so you've got the CTLA blockers, you've got the anti-CD3 mAb, you've got the anti-CD40L mAb, and cyclosporin A, the JAK3 inhibitor. So these are all different sites of action for various agents used, including some of the more general immunosuppressive drugs here. So, just going to check how many slides is left. I can't. That's all right. So, the idea is you want to tolerate the grafts because we want the grafts, right? So, you basically have certain cells and cytokines that are associated with graft tolerance, and we know a key player in tolerance is the Tregs, and the Tregs express FOXP3. So they play a quite important role in transplantation tolerance as well. There are some instances of clinical operational tolerance where the graft survives even if you don't use immunosuppressive therapy, as long as you have the Tregs in the circulation and in the graft, because the Tregs will be the ones to inhibit alloreactive cells, and they'll be the ones producing the immunosuppressive cytokines. And there's also some tolerance that you'll see when you do a transplant in what we call immunologically privileged sites. So these are areas that do not have a very easy access for the immune cells. So if you recall in the earlier slides, a key feature of the rejection is the infiltration of the cells. So the cells all come from the blood vessels and then start attacking in the tissue. So some sites are not as reachable by the blood vessels, or so they cannot have as easy immune cell access. So these are areas like the anterior chamber of the eye, the cornea, the uterus, the testes, and the brain. So when you do transplants there, it doesn't induce an immune response as much because they're basically separated from the cells of the immune system. So this is called physical sequestering of the grafted cells. Yes, so that's actually an approach that they're trying right now to physically sequester the grafted cells for islet cell transplants in diabetes. So if you remember, the islet cells are the ones that get destroyed in diabetes in the pancreas. And then there's also a way to induce the transplantation tolerance. So usually you can induce by having a state of mixed hematopoietic chimerism. So you have the donor and recipient hematopoietic cells coexist in the host even before you introduce the transplantation. So the transplant recipient will undergo a total or transient myeloablative therapy, and then they'll get some bone marrow transferred from the donor before receiving the actual organ that they need. And so that allows a mixture of some of the blood cells from the donor to start existing in the body of the recipient, so that later when the graft from the donor comes in, the recipient is less likely to reject it. So it's developed some tolerance for the organ. Right, I think we're nearing the end now, so this is just a summary. It's quite an old picture. I apologize for that, but just an idea of how many transplants were performed in 2011. You can bet the number has gone up since then. But you can see here cornea. That's basically the one that has the highest number of transplants performed, and most of them come from cadavers. And if you remember why that is, probably because it's one of the more safest or likely to succeed transplants. Remember the whole immune sequestration, so less likely to be rejected. The other one is the hematopoietic stem cell transfer, so the second most frequent transplant performed. Then you have the obviously blood. you can consider that a transplant because it's coming from a donor, although it's not quite the same thing, but blood is actually the most frequent one, then the cornea, and then the kidneys because people need it more. So you actually get a mixture from cadavers, which is bodies that have died, and also the living donors as well. And then you've got a few of these others. So skin grafts, we don't really see that as an allograft. It's mostly autografts because an allograft is very risky for rejection. Great. So now we're at the end. To recap, organ transplantation, basically not something you think of when you talk about immunology, but immunology is such an important thing in the success of transplantation because a lot of the rejection is mediated by the immune response. And we know that it's basically following a set of stages in a set course, and it relates to the specificity and the memory of the immune system. And we know that the T cells play a really key role, although we also know the B cells play a role, especially when we talk about more chronic graft rejections. And so when we want to manage these, we use immunosuppression therapies, and they can be general or they can be specific, or even before that, you can also just irradiate the lymphoid organs to just kill off the T cells. But you do have some T cells that are good for transplants, like Tregs, because they make cytokines that promote tolerance. So we try to harness those to improve translation outcomes. And that's it. So I will see you at the live lecture on Tuesday. Bye-bye. Have a good holiday on Monday.